Put your hands up if you love snakes, if they fascinate you. And who hates snakes? And the reality is that, snakes divide the human population. But because for many years we have lived with snakes, they're intrinsic to our culture. This is in Sri Lanka, where the cobra is a revered snake, because the Lord Buddha was said to have sheltered under the hood of the cobra. And you can see here in Egyptian mythology that Cleopatra killed herself, theoretically, by the bite of an asp. So snake bite and snakes and snake bite is very intrinsic to human culture and human nature. And we find them all over the world. There are almost 3,000 species of snakes, but only about 200-ish really cause severe problems. They are found in tropical and temperate regions, but particularly focused in the tropics. And one of the constant things I'll come back to as I talk is about heterogeneity, variability that you see in snakes. So to give you some ideas, this is the tropical rattlesnake that you find in many bits of South and Central America. This is the kind of snake that you can see why some people are fascinated. A beautiful animal, a Pseudechis species that you see in Australia. This is the biggest venomous snake you find, the king cobra, that may go up to 4 meters long. To give you an example, I recently advised on a person that got bitten because he had put his hand with a mouse inside the snake's mouth to feed it. So it tells you a little bit about how people get bitten by snakes, but also quite how big these things can be. And in contrast, this snake may be only about 12 inches long. It's the carpet viper that you find in the Sahel belt of West Africa, and it probably causes more deaths than any other species. So there's enormous variability in the size, what they look like, and in the venom that they provide. And it's this kind of typical scenario where people get bitten. These are rice farmers in Sri Lanka. The snakes, as you can see from the pictures, would be hidden in that environment. The vast majority of people bitten by snakes are bitten as a part of really an occupational disease, or they're going out to their farms. And it's these poor members of the rural population that get bitten. This is not seen even in relatively poor LMICs. This is seen in the rural areas. It really is the rural workers. And that's to a certain extent why it's been so neglected. This is an estimate of the DALYs, the disability adjusted life years, from all NTDs. So you can see, just for example, this is schistosomiasis, this is hookworm disease, this is rabies. And by WHO estimates, albeit on slightly poor quality data, snakebite is way above that. So this is something that we really do need to address, but has not been addressed for many years. Snakebite is treated by antivenom. Antivenom is very simply a crude product. You get a snake, you take venom out of that snake by milking it, you chuck it into a horse, usually sometimes a sheep, you take the blood out after several months of increasingly tolerizing the animal to that venom, and then you process the blood in some way, and you get antivenom. And what you do to the processing of that blood determines often the safety and the efficacy, because you can actually use affinity purification, you can make it better, but of course it makes it more expensive. And so some snake antivenoms are pretty much unpurified horse serum. Others, for example, used in Australia or in the US, cost lots and lots of money, but they are very, very complex and much safer. The fundamental problem is that putting venom into an animal and getting an immune response does not necessarily mean that you get a good immune response against the toxic components of the venom, because those components that are most toxic to humans may not be very immunogenic, and those components that are very immunogenic may have no effect on humans. And so antivenom is very much a crude product, a very crude product that is not necessarily targeted well against the stuff we want it to be targeted against. And it's been made in the same way pretty much since Pasteur invented it in the 19th century. There have been very few advances. So when we deal with antivenom studies, antivenom trials, we're dealing with something that's not particularly sophisticated. And more importantly, that antivenom is only relevant to the patient that's been bitten by a snake whose venom was used in the production of that antivenom. So that means there are multiple different antivenoms. There's no universal antivenom, and for any one patient, only a certain antivenom may be appropriate. We also see a huge variation in quality. and that means usually, as I said earlier, if you play with it more, it costs more, it gets safer. But safety is a real issue, and there's very little standardization. There is no formal way of dose finding for antivenoms. Most antivenoms have gone into humans without any testing whatsoever, sometimes a little bit of mouse testing. So there are challenges. And that gives you an indication of the kind of rates of adverse events that you can see. I've done a lot of work in Sri Lanka where we commonly see up to 80% of people will have antivenom reactions. And those are antivenoms in general use. So there is a real balance around effectiveness and efficacy and safety to be thought about when we design a clinical trial. It's also fair to say, and this trial, the ANYSNAKES trial, will be focusing on Africa. It's also fair to say that there's been a degree of crisis in the availability of good quality antivenoms. See here, now 8 years ago, highlighted in Nature, but we have still not sorted out that crisis. And so for many patients in sub-Saharan Africa, they do not have access to an effective, safe, or affordable antivenom. And there have been a number of manufacturers that have tried to fill that gap with very poor quality products, including products distributed in Africa that are just not going to work for African snakes. There has been a change, and that's why really, to a certain extent, this is the right time to think about a major antivenom trial. So in 2017, snakebite was designated as a neglected tropical disease formally by the World Health Assembly. And in 2019, there was a new WHO roadmap which includes effective antivenoms as part of the roadmap. And included in that is some formal, for the first time, assessment by WHO of antivenom quality. Now that's done on incomplete data, which may include a little bit of animal data, may include an odd observational study, but for the first time there is an incentive for manufacturers to have some attention to the quality of what they're using, and therefore we do need to evaluate these antivenoms. Because the reality is that the RCTs in snakebite have been incredibly rare, and you can see there over 54 years, when I last looked at this, and it's not changed since 2020, there'd only been 6 RCTs that we consider phase 3. And you can see also where those studies have been. So for example, in Africa, only Nigeria has ever done any RCTs on antivenom. Yet antivenom is used regularly all over the whole continent. There is a gap to fill. So what we would like to do, is to try and conduct a hospital-based, multi-site randomized study in sub-Saharan Africa. And we want to look at both existing antivenoms and the number of new antivenoms that are coming into the market as a consequence of the WHO initiative to develop new antivenoms. And fundamentally, try and develop the scientific evidence that helps somebody sitting in the Ministry of Health decide, I need to get that antivenom, rather than one that's being sold to me without any efficacy data, as is currently the case. As I've said, there are a few challenges in undertaking such a study. So first of all, not all antivenoms work against all species. And within the range of antivenoms that will become available in Africa, not all will have the same neutralization efficacy against all species, because it depends what venom has been used in the manufacture of that antivenom. We also see, even within countries, a huge difference. In the north of Mali, for example, the snakes you see are completely different to the snakes you see in the south of Mali. And they will produce, the different species will produce different clinical syndromes and different symptoms and signs. It also follows that you can have the same clinical syndrome as a consequence of being bitten by 2 or 3 species, different species. So you cannot use the clinical signs in a patient to determine what species has bitten that individual. And then when anyone is bitten by a specific species, the amount of venom that goes into them is highly variable. If a snake has just fed and it's put all of its venom into its prey, then you can be bitten by a highly venomous snake and have no clinical problems whatsoever. Equally, the other way around, if it's not fed for weeks, you may get a large amount of venom going into your individual and die immediately. I've treated patients who have come in and they've been out hunting, their dog has been bitten and the dog has died, and they've come in without any problems. And that's almost certainly because most of the venom has gone into the dog. But that means clinically, yet again, there's heterogeneity in what you see. Just because you've been bitten by a particular snake doesn't mean you see a clinical syndrome. And then the final thing is to comment on is that of the antivenoms currently in use, hardly any have any clinical data of note. Certainly no RCT, with one exception, no RCT evidence to guide how good the antivenom is. But there are existing antivenoms that we would want to put into a trial without any clinical data to get to at all, including on the right dose. So, what are the challenges we want to take on in this trial? So, first of all, the existing antivenoms and the novel antivenoms that we anticipate are coming through will vary in their suitability for any particular species. And we don't know what the right dose of those antivenoms will be, because dose-finding studies are not commonly done, and they're also challenging because of the heterogeneity. You need large numbers, essentially because of heterogeneity. And as we've heard, why is a practical design appropriate here? Because there's certainly no single standard of care for an individual sitting in a particular location. It's also very unlikely that one single antivenom will be the optimum antivenom across the whole of sub-Saharan Africa because of the change, or because of the change in distribution of species and the way that antivenoms are made up. And so we certainly couldn't have a single comparator across the whole of sub-Saharan Africa. We also do need a trial design that will allow us to actually incorporate new antivenoms as they come onto the market. We anticipate in the next 5 years there are at least 3 or 4 new antivenoms made to much higher quality standards that will come into the market. And we would also want to get rid of ones that are not doing very well. We're going to focus on 2 main clinical syndromes, and those are coagulopathy and shock. They will, to a certain extent, overlap, but we're going to treat them separately for the main analysis because it's quite possible that a single antivenom may be better against the coagulopathy than it is against shock, for example. And it's very similar to the example we've seen with Neosep. Each participant will be randomized to a set of regimens or antivenom regimens that's appropriate to a particular clinical scenario in that site. So it's not done on an individual sensitivity basis, but it is done on the basis of actually, in that particular geographical site, there are a certain number of species that will cause that clinical syndrome, and therefore you are randomized to that set, the antivenoms that will deal with those species. We're also very interested, really, not in saying antivenom B is better than A or C is better than D, but in looking at which antivenoms reach a certain threshold of efficacy and of safety. And again, as we've heard already, ranking those antivenoms and so therefore giving policymakers a list of antivenoms of a certain quality, and then they can decide in their particular scenario, geographical location, which of those is better for them to obtain. This is a similar graph to what you saw with the ESF, but for example, in site 1 for coagulopathy, A, B, and C may be appropriate antivenoms, but only A and B contain the venoms or were made using venoms of snakes that cause shock, for example. And so we're going to have that kind of complexity in the randomization sequences that we use for any one site. And it's also possible that in one site, for example in shock, at site 3 or site 4, there's only one antivenom that's suitable for an individual to be randomized to. The other challenge, of course, is, as I said already, that we have very little data and some challenges in doing dose optimization. So we will certainly incorporate this into the study as well. And basically, we'll determine a minimum and a maximum dose of antivenom that we think is appropriate for that individual for that particular clinical syndrome, and then randomize people to both a specific antivenom on the basis of a site list and to a specific dose of that antivenom within the minimum maximum that we think is appropriate. We will get rid of antivenoms that are not doing well either on the efficacy data or more likely, I think, on safety data. There's a safety threshold over which we think it's unacceptable for antivenom to be used. And the sample size has been calculated by Becky very much as she just described, using for coagulopathy and shock separately, using the approach she described. And we're going to look for around 1,000 patients for coagulopathy and 1,000 patients of shock. I'm not going to talk about outcome measures a great deal, just other than to say the collaboration came out of work of a number of people getting together to develop core outcomes for snakebite clinical trials. And again, they reflect the heterogeneity in that there are some universal to mortality, disability reactions, either acute or chronic. And then there are syndrome-specific core outcomes. And the ones that we're focusing on this trial are the resolution of coagulopathy measured by an INR less than 3 to 8 hours, and then the resolution of shock with a blood pressure at 3 hours. But there are a number of different core outcomes that we've developed for different syndromes and different species around the world. So finally, this application has been submitted to the Wellcome Trust. This is not yet funded, and it is a challenge to get this funded. It's going to be a challenging study in that almost every site we use will be effectively a site that's not done clinical trials before, by the very nature of this being a disease that affects people in rural populations and remote areas. And so we're, again, a little bit like Mike talked about, we're going to try and do a complex study in sites that have never done trials before. I'm very heartened to hear his discussions and experience with the regulatory authorities in South Africa. One of my biggest concerns has been how we managed to do this. And AVRF, which I don't know whether people are aware of, but it's an association that essentially is an umbrella organization for ethics and regulatory frameworks in Africa. We've had some preliminary advice from them, and what they often do is give some approval with expert input, which then allows other individual country regulatory authorities or IRBs to give approval without having to go through a complex process. And we hope they will help us with that. And we will know in the next couple of months if this is going to be successful. So thank you.